Magnetic modulation biosensing, or MMB, is a novel technique for the detection of biological molecules. Here, a proof of principle assay is shown in which interleukin-8 is detected. When interleukin-8, also called IL-8, is added to a reaction mixture containing antibody-coated magnetic beads, biotinylated anti-IL-8 antibody, and fluorescently labeled streptavidin, a molecular sandwich is created, effectively linking the fluorescent molecule to the magnetic bead. The reaction is placed into a cuvette, and alternating magnetic field gradients are applied through two electromagnetic poles, which maneuver the beads into oscillatory motion. The periodic motion of the bead-attached fluorescent proteins in and out of an orthogonal laser beam produces a fluorescent signal that is collected by a PMT and demodulated using a lock-in amplifier. The movement of the beads can be visualized using a CCD camera in place of the PMT. Synchronous movement is seen in samples where the magnetic beads have been linked to fluorescent proteins via IL-8.
Hi, I'm Arie from the Nonlinear Optics and Laser Lab at the Department of Physical Electronics, Tel-Aviv University.
I'm Noga Porat from the Department of Biological Sciences in the University of Illinois at Chicago.
And I'm Marcelo Ehrlich from the Cell Research and Immunology Department of the Tel-Aviv University.
Today, we will show you a method for rapid and homogeneous detection of biological assays using a magnetic modulation biosensing system.
We are using this procedure in our laboratory to detect specific proteins and DNA sequences at low concentrations.
So let's get started.
Begin this procedure by setting up and labeling a tube for each reaction, including a target sample and a non-target control sample. To each tube,